And so now we're going to move on to the next portion of gene expression, which is protein synthesis or translation. And so once we have a gene that has been transcribed, we're going to convert it into, through translation, cells are going to convert it into a polypeptide or a protein. And traditionally, one gene will only be able to encode one polypeptide or one protein. And the polypeptide that gene encodes is determined by the mRNA codons. And codons are just triplets of nucleotides, and you can see them here, right? So one codon would be UUU. And each triplet of amino acids, or each codon, encodes one particular amino acid, right? So the codon UUU, or uracil, 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 would encode the amino acid phenylalanine. And this is what's called the genetic code table or the codon table, and it helps you translate, pun intended, the codons of mRNA into which amino acid they encode. And so there are 20 different amino acids, and I'm not going to ask you to know them or memorize them for this particular class, but it is important to realize that some of the amino acids have common properties. Some are hydrophobic or water-fearing, some are acidic, some are charged, and the chemistry of the amino acids is determined by this functional group, or this R group, which is variable between all of the different amino acids, and that's what confers these chemical properties to them. And the chemical properties within the amino acids of the protein give the protein some of its functions and structure as well. So in order to turn an mRNA into a protein via translation, we need a couple players, right? So we need the mRNA that's going to be translated, and we already talked about how that's made via transcription. But in addition to that sort of instruction code in the mRNA, we also need an adapter that can take the mRNA codons and basically bridge the gap and add the correct amino acids to the correct codon. And so the adapter between amino acids and mRNA is what's known as a tRNA. And it's named a tRNA, one, because it transfers amino acids to a growing polypeptide, and also because it's shaped sort of like a T. And there are two parts to a tRNA that are important. The first is the anticodon, which is basically a sequence that's complementary to the codon in mRNA, and then a corresponding amino acid. And so what the tRNA can do is it can bind to this complementary region in the mRNA and have the correct amino acid attached in that location to build the polypeptide. And there's a special enzyme known as aminoacyl tRNA synthase that actually ensures that the anticodon on that tRNA and that tRNA has the correct amino acid attached to it. And so aminoacyl tRNA synthase adds the amino acids to the tRNAs, and then the tRNAs can bring these amino acids to the ribosome and bind to the mRNA and help create the polypeptide. And additionally, the enzyme that really does translation is the ribosome. Ribosomes are here to make protein. Eukaryotic ribosomes are made up of two subunits, a large subunit, or the 60S subunit, and a small subunit, or the 40S subunit. These ribosomes are made up of different types of RNA, and this RNA can have several functions. First of all, it makes up the structure of the ribosomes themselves, but there are also different types of RNA that are involved in initiating translation or protein synthesis, as well as catalyzing the formation of the peptide bonds, or the bonds that form between the peptides or the amino acids to create a polypeptide. And so ribosomes are, for all intents and purposes, the enzymes that complete translation. And so you have the mRNA, which is your instructions. You have the tRNA, which is bringing those amino acid pieces to the ribosome, and then the ribosome, which is going to build that protein using the mRNA instructions. And there are specific sites on the ribosome where this happens. And so the first is what's known as the amino acyl site, or the A site, which you can see right here. And that's where an incoming tRNA binds and brings the next amino acid that's going to be put onto the growing protein. There's the P site, or the peptidyl site, where the peptide bond is formed, and that binds to the tRNA to which the growing polypeptide is attached. And then there's the E site, or the exit site. And the exit site binds to a departing tRNA. And so the A site binds to an incoming tRNA carrying the next amino acid. The P site is where that is the site that binds to the tRNA containing the growing polypeptide. And the E site is the site that binds to the departing or the leaving tRNA that's already donated its amino acid to the growing polypeptide and is about to move out. And protein synthesis moves in the same direction as DNA replication and transcription from 5' to 3'. And the ribosome will move down the mRNA instructions in a 5' to 3' direction, synthesizing the polypeptide as it goes. And the way that works is by having an incoming tRNA with an anticodon that's complementary to the mRNA here, bind into the A site, which is empty, and it brings the correctly corresponding amino acid to that codon that was in mRNA, right? And then it can transfer the growing polypeptide that's here is transferred to this tRNA, and then the ribosome shifts slightly. 3 nucleotides down, and this orange tRNA, which was originally in the P site, holding the growing polypeptide chain, when the ribosome shifts, ends up in the E site and is ejected. And now this purple tRNA is in the P site, ready to transfer the growing polypeptide chain to the next incoming tRNA that will enter the A site. Right, and so you have this orange tRNA at the P site and a growing polypeptide chain on it. Both the E and A sites are empty. The correct tRNA will come in and bind to that A site based on the anticodon it has here. As long as it matches the codon in the mRNA, we're good. It brings the correct amino acid that should be added next in sequence. And then the growing polypeptide chain is transferred to that tRNA at the A site. The ribosome shifts over one codon, and then that orange tRNA ends up in the E site and is ejected, and the A site becomes available for the next amino acid. And this continues all the way down from the 5' end of the mRNA to the 3' end until the whole mRNA has been translated into a polypeptide. And then to end translation, there is a stop codon, and the stop codon tRNA actually doesn't contain an amino acid, but rather it contains a water. And so rather than adding that polypeptide or adding an amino acid to that chain, the stop codon tRNA will basically chop off the polypeptide, free it, and then the ribosome will dissociate from the mRNA and the tRNAs and can synthesize another protein. And so translating the protein is only the first step in terms of creating a functional protein, right? So first we have to take the mRNA sequence and convert it into amino acids to create this polypeptide. And that would be considered a primary protein structure you can see here. Literally just the sequence of amino acids, sort of this linear shape. But proteins, in order to function, have to be folded into what's known as their native structure. And those folding instructions, luckily, are encoded in the amino acids of the primary sequence as well. And so once the primary sequence or this protein has been synthesized by the ribosome, it takes on what's known as the secondary structure, which is there are some common ones known as beta-pleated sheets and alpha helices. And then it takes on a more 3D conformational structure known as tertiary structure. And sometimes there are multiple proteins that come together to make a quaternary structure. And I often think of this in terms of origami, where the primary protein structure is the piece of paper. The first couple folds would be considered the secondary structure. Still doesn't really look like anything. You still can't tell what animal you're folding in terms of the origami. But then once the origami takes the shape of a swan, for example, it's achieved a tertiary structure or a 3D structure that's recognizable. And then a group of swans would be considered quaternary structure. Not all proteins will have quaternary structure. Some will just have up to tertiary. But what's important to remember is that this process is not exactly simple. Some protein structures are very complicated. And the cytoplasm has a lot of other stuff going on, right? There's other molecules in there. There's a possibility that this polypeptide can interact with other things. And so there's a specific molecule known as a molecular chaperone that helps proteins fold. And it can do that in two different ways. It can do it by preventing the misfolding of the protein in the first place, and it can also help to correct misfolding of proteins. And so there are many different types of chaperones, and depending on which chaperone you're talking about, it may have a different function. Some of them are passive, just sort of creating a cage around a protein that's folding to prevent it from interacting with other things. Some of them are active. They can make bonds and break them. They can correct misfolding in this way. But they're really necessary because not only does the protein need to be folded in order to be functional, bad things can happen if a protein is misfolded. Because not only can it be non-functional, meaning your cell doesn't have the protein that it needs, it can also gain a toxicity or a toxic function and start to cause damage to your cells. And so protein folding as a sort of final step is very important kind of post-translational process.